Welcome to this video lecture, that is receptor mediated endocytosis. So before going into this receptor mediated endocytosis, let us know what is endocytosis. So endocytosis is a general term for the various type of active transport that move particles into a cell by enclosing them in a vesicle made out of the plasma membrane. There are variations of endocytosis, but all follow the same basic process. First, the plasma membrane of the cell invaginates, that means folds inward, forming a pocket around the target particle or particles. The pocket then pinches off with the help of specialized proteins, leaving the particles trapped in a newly created vesicle or vacuole inside the cell. Endocytosis can be further subdivided into phagocytosis, pinocytosis, and receptor mediated endocytosis. So, there are three pathways by which these molecules they move into the lysosome. So these are phagocytosis, endocytosis, and autophagy. Through the process of endocytosis, these molecules they move through early endosome, late endosome, and to lysosome. Similarly, in phagocytosis, these bacteria they can get entrapped inside the phagosome and ultimately to this lysosome. Similarly, these fragmented organelles they can be entrapped inside autophagosome and ultimately they move to this lysosome. So these are the three pathway how things come to lysosome, where it is fragmented or say digested. Now in endocytosis, I told there is pinocytosis and receptor mediated endocytosis, and pinocytosis is called cellular drinking, and here through this phagocytosis and through the endocytosis and this autophagy, the molecules they move to this lysosome. These phagosome or this endosome or this autophagosome, these are produced from this plasma membrane. So this is an overview of pinocytosis, by which the fluid molecules they move inside the cell up to the lysosome. So here this is the invaginated plasma membrane, to which there is a proteinaceous coat. So this kind of structure in the plasma membrane is called peat, and when peat is coated with a proteinaceous coat, we call this as a coated peat. So this coated peat is pinched up, and then these coat molecules or proteinaceous coat is released, and this early endosome and then late endosome are formed, and during that process the molecule is digested. See, there are two terms here, one is early endosome, another one is late endosome. It is because the early endosomes are formed within 1 minute of the endocytosis, and here the pH is around 6, but in case of late endosome the pH is around 5 to 6, 5.5 to 6, and these are observed after 5 minutes, and then these late endosome they merge with this lysosome, which are found after 30 minutes. Here the pH is less than 5. Okay, so here these molecules or these foreign bodies, they are digested. So here this is the electron microscope view of coated pit or coated vesicles. These are coated vesicles, these are coated pit, and this is the coat protein. Now this coat protein is made up of clathrin. This clathrin has a triskelion structure composed of 3 heavy chains and 3 light chains. These are these light chains, whereas these are the heavy chains. So these heavy chains have the molecular weight around 180 to 190 kilodalton, and these clathrin light chains are 40 kilodalton of mass, and they form a peculiar structure like geodesic clathrin cage, and that is found over this vesicle, that is called your coated vesicle. Now this clathrin coated vesicle has several components. This is the clathrin coat that is attached to this vesicle by one protein that is called adaptin, and this adaptin is attached to the receptor to which the cargo molecules are attached. So this is the complete structure of a clathrin coated vesicle. So these adaptins are adapters that bind clathrin and cargo receptor, thereby regulating which cargo gets loaded into clathrin coated vesicle. Then once these clathrin coated vesicles are formed, they are still before the formation of this clathrin coated vesicle, these clathrin coats here, now it is attached to this pit. So now this is called a coated pit, and before the formation of this clathrin coated vesicle, this invagination grows further and this forms a racket like structure, and to the handle of this racket like structure there is another protein that is called dynamine. So the dynamine is a protein that helps in pinching of this coated vesicle from this plasma membrane. So the dynamine gets energy from GTP. So this is an GTP analog. So once this coated vesicle is pinched off from the plasma membrane there is uncoating of this clathrin proteins and naked transport vesicle is produced. Now, I told you that this dynamine is a GT page. So, how it is known? Whenever GTP analogues are used in place of GTP, this dynamine could not bring the pinching of the vesicle. It is because the non-hydrolyzable GTP analogues block the endocytosis process. So from this it is known that this dynamine is a GTP analog, because this dynamine breaks the GTP to GDP and inorganic phosphate, and the energy is utilized in pinching of the vesicle. But when there is non-hydrolyzable GTP analog, this is not cleaved by this dynamine or hydrolyzed by this dynamine. As a result, there is no energy that can be utilized for pinching of the vesicle. As a result, this process of endocytosis is stopped. Now, these clathrin coated vesicles are rapidly uncoated. Now here you see this dynamine is here, and with the utilization of GTP and breakdown of this GTP to GDP and inorganic phosphate, this clathrin coated vesicle is pinched off, and again with this hydrolysis of ATP, that is ADP and inorganic phosphate, what happens? The naked transport vesicle is formed. Okay, so clathrin coated ATPase is this ATP is hydrolyzed by this clathrin coated ATPase, that is a member of heat suck protein 70 family of chaperones, and it requires ATP hydrolysis. Now these naked transport vesicle, they are targeted to this endosome, and the clathrin and adaptins are recycled. Now, how do cell take up specific macromolecules? So you will see one of the example of a receptor mediated endocytosis. For example, this low density lipoprotein, how this cholesterol is released through the process of receptor mediated endocytosis. We will take up this as a specific example. This low density lipoprotein or LDL is a macromolecule. How this is formed? This is composed of around 800 phospholipids, 500 molecules, around 500 molecules of cholesterol, 1500 molecules of cholesterol ester, and 1 copy of apoprotein B. And the total mass of this LDL, that is low density lipoprotein, is around 3000 kilodalton. So these are the LDL receptors, those are found over the plasma membrane. these LDL receptors, now you can see this LDL receptor over this coated pit, and this is this low density lipoprotein. So this low density lipoprotein is recognized by this LDL receptor, and this LDL receptor brings this LDL to this clathrin coated vesicle, and this clathrin coated vesicle, after being uncoated, fused with the endosome, and it is transported or transferred to the lysosome, and after the action of these hydrolytic enzymes at lysosome, free cholesterol is produced, and the receptors, that is the LDL receptors, now they move back to the plasma membrane for the next round of action. So low pH of endosome causes LDL to dissociate it from a receptor, and the LDL is transferred to lysosome after being fused of the vesicle from trans-Golgi network. Then hydrolytic enzymes cleave LDL and release cholesterol to cytoplasm for continued membrane biosynthesis in smooth endoplasmic reticulum. The receptor is recycled back to surface, and the cycles happens about in every 10 minutes. Now, when there is defect in LDL endocytosis, we suffer from diseases like hypercholesterolemia. So in severe atherosclerosis at early age, that is responsible for strokes and heart attacks in preteens, and excess of LDL in circulating blood, LDL not properly internalized by cells, recessive, this LDL is not internalized because due to mutation in this single gene encoding for plasma membrane receptor for LDL. So this receptor is a protein that is coded by a single gene, and when there is a mutation which is recessive, so when there is a recessive mutation is recessive, and when it is there is a defective LDL receptor. As a result, this LDL is not internalized. As a result, we suffer from the diseases, and these diseases provide insight to mechanism of receptor mediated endocytosis and identification or functioning of LDL receptor. So whenever there is a mutation in the N-terminal domain of this LDL receptor, then what happens? The receptor, the LDL receptor, doesn't bind to low density lipoprotein. It is because when there is a mutation, the amino acid sequence is changed. As a result, this LDL is not compatible with this receptor. As a result, there is no binding of the receptor to this LDL. Now whenever there is a mutation in C-terminal domain or this carboxyl terminal domain, what happens? The LDL receptor is not internalized. So even if the N-terminal domain is perfect in the receptor, but due to defect in the C-terminal domain or changes in the amino acid sequence in the C-terminal domain, the LDL receptor is not internalized. So even if the LDL is recognized by the LDL receptor, it cannot come inside this cell. So as a result, there is excess LDL in circulating blood, and this LDL is not properly internalized. So whenever there is a mutation either at C-terminal end or at N-terminal end, there is a defect in LDL endocytosis. Now we are talking regarding this domain in the LDL receptor. So this is the N-terminal end of this LDL receptor. That N-terminus of the LDL receptor binds apoprotein B in LDL. Now this is your LDL, and this is LDL receptor, and this is the C-terminal. This is the N-terminal end, and this is the C-terminal end. So in the C-terminal end, there is asparagine, proline, valine, and tyrosine, and it binds with the adaptin complex, because this adaptin complex will bind with the clathrin molecules, because without the help of clathrin molecules, these LDL cannot be internalized. So whenever there is a mutation in this region, this cannot bind with the adaptin, so also this clathrin. And when there is no binding of adaptin or clathrin, this cannot move inside this cell. So this adaptin complex bind to endocytosis signal in cytoplasmic domain of receptor. So there are at least 3 different adaptin complexes that bind different endocytosis signal on receptor, and this adaptin recruit clathrin and initiated coated pit and vesicle formation. So whenever there is a defect in, or there is a change in the amino acid sequence of this C-terminal end, then this LDL cannot be internalized. Now a single coated pit, it can have many different receptor, so also it can carry different cargo molecules. So here is one kind of cargo molecule, this is another kind of cargo molecule, and this is also another kind of cargo molecules. And in a coated pit, there are around 1000 receptors, and some coated pit used for this receptor mediated endocytosis, some are used for pinocytosis. Now the outside the cell, on the outside of the cell, there the pH is around 7 to 7.2 in normal condition. Now once these receptors are present over the plasma membrane, and there is invagination in the plasma membrane called the pit. So plasma membrane is not uniform at all places, there are some thick and thin places, and these are called your pits. So to these pits, these receptors are attached, and these receptors, they can recognize these ligand molecules, and these receptors are attached to this clathrin molecule with the help of one protein that is called adaptin, and this coated pit, the invagination of the coated pit increases further, and with the help of dynamin, that is 1-GTPase, it is pinched off from the plasma membrane to form a coated vesicle. Now this coated vesicle is uncoated. So this coated vesicle is uncoated by uncoating protein, that is HSP70 family of chaperones, and that is 1-ATPase, which hydrolyzes ATP to ADP and inorganic phosphate, and that energy is utilized in dissociating the adaptin complex, that is adaptin and this clathrin, from this coated vesicle. As a result, a naked vesicle is formed. So these naked vesicles, they are fused with these early endosomes, which are formed within 1 minute of endocytosis, and the pH is around 6, and these early endosome, they get converted to late endosome, where the pH is again decrease, like from 5.5 to 6, which again merge with this lysosome. So this proton pump in endosome acidifies endosome lumen, and this cause LDL to dissociate from the receptor. Now in the endosome, the acidic nature help in the dissociation of receptor and LDL from the receptor. Now these LDL moves into this lysosome, and these receptor, dissociated receptor, they again move through this move to this plasma membrane for another cycle of endocytosis. Now in this lysosome, from the LDL, the cholesterol is freed, and the cholesterol ester is cleaved, and the cholesterol is released for used for the formation of different membrane or membrane like things. Now when there is a defect in receptor mediated endocytosis, we suffer from heart related syndromes. Though receptor mediated endocytosis is intended to bring useful substances into the cell, other less friendly particles may gain entry by the same route. For example, these flu viruses, diphtheria and cholera toxin all use receptor mediated endocytosis pathway to gain entry into the cell. Suppose a certain type of molecule were removed from the blood by receptor mediated endocytosis, then there will be certain problem with the functioning of the cell. So sometimes this receptor mediated endocytosis is helpful in providing useful molecule to the cell, but at certain times it is also disadvantageous for the cell, as I was telling you regarding the entry of different kinds of viruses. So, receptor mediated endocytosis is a form of endocytosis in which receptor proteins on the cell surface are used to capture a specific target molecule. These receptors, which are transmembrane proteins, cluster in region of the plasma membrane known as coated pits, and when the receptor bind to their surface by the specific target molecules, endocytosis is triggered and the receptor and their attached molecules are taken into the cell in a vesicle, and these vesicles are coated by certain proteins. We call them as clathrin molecules, and they take these proteins or these cargo molecules along with the receptor inside the cell, where the different phenomenon occurs, and these molecules are released inside the cell after the digestion or after certain processes at the lysosome. So, this endocytosis is not only helpful for getting substances inside the cell, rather it also helps in killing different bacteria, and those are coming to the cell, and in sometimes it is also disadvantageous in taking these viral particle into the cell along with these cargo molecules. So, this is all about receptor mediated endocytosis, how these are advantageous and disadvantageous for the cell, and how the mechanism occurs, how these cargo molecules they are taken inside the cell, and how they are digested, and how the receptors are again taken back to this plasma membrane. We have discussed in detail. So, you can expect some of the question like how the process of receptor mediated endocytosis occurs, describe its mechanism, or you can expect a question like how this cholesterol is released for use in a cell through the process of receptor mediated endocytosis. And you can get objective questions like what is the composition of this LDL receptor, or what is the composition of this low density lipoprotein, what are the sequence of amino acid at the C terminal domain of this LDL receptor, why dynamine is essential for receptor mediated endocytosis, so and so. So, you can check the answer for this question, and thank you for listening.